You are walking under a canopy. The path is soft underfoot. The air smells different from the street you left 20 minutes ago. Something green, something resinous, something the lungs draw in without conscious effort. The shoulders dropped 10 minutes ago. The jaw unclenched. The breathing slowed from the urban pace to something deeper, each breath pulling more air from the lower branches where the concentration of volatile compounds is highest. 2 hours pass. You return to the car. The muscles feel the same post-walk looseness as any other 2-hour walk. The mind feels clearer than it should from the distance covered. The walk felt calming in a way the neighborhood walk does not, a calm that persists through the evening and into the following day. You attribute the calm to the scenery, the quiet, the break from screens. 7 days later, a blood draw shows the difference. The immune cells responsible for destroying virus-infected cells and early-stage tumor cells are 50% more active than before the walk. The same walk through an urban environment, same duration, same intensity, same distance, produced zero measurable change. The urban air contained exhaust particulates, ozone, nitrogen dioxide, but no terpenes. The lungs processed an identical volume of air. The immune cells showed no response. The difference between the two walks was invisible, odorless at conscious concentration, and measurable only through laboratory analysis of the blood drawn a week later. The variable was the air. The molecules in the air were the only difference, and those molecules were still changing the blood a full week after the last breath of forest air. The air under the canopy contained volatile compounds the trees were releasing as part of their own defense system, chemical warfare against fungi and insects. You walked through the tree's immune system. The molecules crossed from the lungs into the blood. The blood carried them to the natural killer cells. The natural killer cells upregulated the genes that encode the proteins they use to destroy infected and malignant targets. The upregulation persisted for 7 days from a single 2-hour exposure. The benefit was molecular. The calm was a side effect. The enhancement was operating below the threshold of conscious awareness. You experienced the walk as calming. Your blood experienced the walk as an immune intervention measurable a week later. What are the molecules, and why are the trees releasing them? Terpenes. Isoprene-derived hydrocarbons produced by every tree species, released continuously through leaf stomata and bark into the surrounding air. Alpha-pinene, the molecule that gives pine forests their characteristic scent. A bicyclic monoterpene, molecular formula. C10H16, molecular weight, 136 grams per mole. D-limonene from deciduous species. Beta-pinene, camphene, myrcene, 1,8-cineol. Concentrations in temperate forest air 2 to 20 parts per billion, higher in summer when temperature-dependent volatility increases, highest in dense coniferous forests under closed canopy where the molecules accumulate before dispersal. A summer afternoon under a dense pine canopy delivers the highest terpene dose. A winter walk through a bare deciduous forest delivers the lowest. The season determines the dose because temperature determines the volatility, and volatility determines how many molecules leave the leaf surface and enter the air you breathe. Terpene concentration varies by species. Conifers release alpha-pinene and beta-pinene at concentrations 3 to 5 times higher than deciduous species in the same forest. Canopy density matters. A closed canopy traps terpenes beneath it, building concentration the way a ceiling traps heat. Time of day matters. Afternoon concentrations exceed morning because temperature-driven volatility peaks with solar heating. The practical consequence, if you live near coniferous forest, the terpene benefit is available year-round because conifers retain their needles and continue releasing terpenes in winter, though at reduced concentrations. Trees produce terpenes continuously as antimicrobial agents that kill fungi on direct contact, as insect deterrents that repel herbivores, and as inter-tree signaling molecules that warn neighboring trees of insect attack. The molecules are the tree's immune system, chemical weapons deployed into the air continuously, part of a defense strategy that evolved hundreds of millions of years before mammals existed. You walk through the crossfire of a chemical war between trees and fungi, and the weapons happen to activate a human immune pathway through cross-kingdom molecular compatibility that neither kingdom evolved to exploit. The molecular compatibility is ancient. Terpenes interact with cellular signaling pathways that are conserved across kingdoms because the fundamental cell biology they target, membrane function, intracellular signaling cascades, gene regulation, has been conserved since the common ancestor of plants and animals diverged over 1 billion years ago. The compatibility is accidental and ancient. The tree did not evolve terpenes to help humans. Humans did not evolve NK cells to respond to terpenes. The interaction works because the molecular targets the terpenes evolved to hit in fungi happen to overlap with signaling pathways inside human immune cells that regulate gene expression. The overlap is a billion-year-old coincidence that produces a measurable benefit neither kingdom designed. The tree's antifungal weapon works on human immune cells because the cellular machinery the weapon targets is old enough to predate the divergence. Molecular weight matters. At 136 grams per mole, alpha-pinene is small enough for passive diffusion across the alveolar membrane in the lungs. No active transport required. No receptor needed. Fick's law of diffusion. The molecule crosses the membrane driven by its concentration gradient from the higher concentration in the inhaled air to the lower concentration in the pulmonary capillary blood. Within minutes of the first breath under the canopy, terpene molecules are circulating in the bloodstream. Every subsequent breath adds to the concentration. The diffusion rate follows Fick's law, proportional to the concentration gradient across the membrane and the surface area of the membrane. The alveolar surface area in an adult lung spans approximately 70 square meters, the largest gas exchange surface in the body, and every square meter is available for terpene diffusion during every breath. The surface area that evolved to absorb oxygen absorbs the terpenes through identical passive physics. Each breath cycles approximately 500 milliliters of air across this surface. At a breathing rate of 12 to 15 breaths per minute during a walk, the lungs process 6 to 7.5 liters of terpene-containing air per minute. Across 2 hours, 720 to 900 liters of forest air passing across 70 square meters of absorptive surface. The cumulative exposure is substantial even at parts per billion concentrations, because the volume of air processed and the surface area available for absorption are both enormous. The exposure of breathing forest air produces a measurable serum terpene level that persists as the molecules distribute into tissues throughout the body. The absorption is passive, continuous, and cumulative. Every breath adds its increment to the circulating pool, and the pool builds across the full duration of the forest exposure without requiring any conscious effort beyond breathing. Lipophilicity matters. Terpenes are hydrocarbons, fat-soluble molecules that cross cell membranes as readily as they cross the alveolar membrane. When circulating terpenes encounter a natural killer cell, they enter the cell through identical passive diffusion that brought them into the blood. No receptor, no signal. The molecule walks through the cell membrane because the membrane is a lipid bilayer and the molecule is lipophilic. The terpene enters the NK cell the way oil enters butter. The chemistry of the molecule matches the chemistry of the barrier. No enzyme activates the entry. No energy is consumed. The molecule simply dissolves through the membrane driven by the concentration gradient between the bloodstream and the cell interior. The same lipophilicity that allows terpenes to cross the alveolar membrane allows them to cross every cell membrane they encounter. The NK cell receives the terpene because every cell in the body receives it. The NK cell is the one that responds by upregulating its killing machinery. Molecules enter the blood. The molecules enter the NK cells. The question is what they do inside the cell that changes the killing efficiency for 7 days. What happens inside the NK cell after the terpene enters? Natural killer cells patrol the bloodstream, scanning every cell they encounter for a molecular identity badge. Major histocompatibility complex class 1, a surface protein healthy cells display. Virus-infected cells and cells undergoing malignant transformation frequently lose their major histocompatibility complex class 1 badge. The missing badge triggers NK cell activation. The detection system is elegant in its simplicity. The NK cell does not need to recognize the specific virus or the specific cancer. It needs to detect the absence of the identity badge healthy cells display. Any cell that has lost its badge for any reason activates the NK cell. The system catches threats the adaptive immune system has never encountered because the detection mechanism does not require exposure to the threat. Every hour of every day, NK cells circulate through the bloodstream, the spleen, and the lymph nodes, scanning thousands of cells for the missing badge. The surveillance is continuous and autonomous, requiring no instruction from the adaptive immune system and no prior encounter with the specific virus or malignancy. The terpene enhancement makes each patrol cell more lethal per encounter, widening the margin between detection and destruction. The activated NK cell binds the target and releases cytotoxic granules. Each granule contains two killing proteins. Perforin polymerizes on the target cell's membrane, assembling into ring structures approximately 16 nanometers in diameter that punch pores through the membrane, holes large enough for the granzyme enzymes to pass through. The pore is a molecular gate. Perforin assembles the gate, granzyme walks through it. Without sufficient perforin, too few pores form and too little granzyme enters. The terpene enhancement of perforin production directly increases the number of pores each NK cell can punch per engagement, which directly increases the granzyme delivery, which directly increases the killing efficiency. Granzyme enzymes enter through the perforin pores and activate the cell's internal self-destruction program, apoptosis, triggering the cascade that dismantles the cell from the inside. The target dies. The NK cell detaches and resumes patrol. Each NK cell carries approximately 200 granules. The killing capacity is finite per engagement, but renewable. The cell resynthesizes perforin and granzyme over 24 to 48 hours. Terpenes entering the NK cell upregulate the genes encoding perforin, granzyme A, and granzyme B. A factory receiving a rush order to retool its assembly line, the terpene molecule is the work order. The gene expression machinery is the factory floor. The perforin and granzyme are the products. The factory runs the order, produces the weapons, stocks them in granules, and the granules remain loaded long after the work order expired. The Saturday morning walk where you felt nothing beyond the calm was the morning the factory floor inside every circulating NK cell received its work order and began manufacturing weapons you would carry, unfelt, through the following Friday. The gene expression change is the mechanism. The terpene molecules interact with intracellular signaling pathways that increase the transcription rate of the perforin and granzyme genes, producing more messenger RNA, which the ribosomes translate into more protein, which the cell packages into more granules with higher killing protein content per granule. Each NK cell becomes a more effective killer because each granule carries more perforin to punch more pores and more granzyme to trigger more apoptosis. The enhancement is quantitative. Lyse studies measured the intracellular concentrations of perforin, granzyme A, and granzyme B individually, confirming that all three killing proteins increased measurably in the NK cells of subjects who walked in the forest. The cells were not simply more numerous. Each individual cell was better armed. The distinction matters for immune surveillance. A higher number of NK cells increases the probability that a patrol cell encounters an abnormal target. A higher killing efficiency per cell increases the probability that the encounter results in target destruction. The terpene exposure improved both variables simultaneously, widening both the coverage and the lethality of the immune patrol. 7-day persistence is the gene expression half-life. The proteins manufactured during the terpene exposure are already synthesized and stored in granules. The granules remain functional for days to weeks. The upregulated gene expression declines gradually after the terpene concentration drops, but the proteins already produced persist in the granules long after the molecules that triggered their production have cleared from the blood. The blood draw at the week-long window is measuring the stored output of a gene expression event that occurred during the walk. The persistence makes biological sense. Gene expression changes that upregulate protein production fill the cell's granule inventory. The granules, once loaded with perforin and granzyme, remain functional until the cell uses them or the proteins degrade through their natural half-life. The protein half-life in cytolytic granules is measured in days to weeks, long enough for the enhanced inventory to persist well beyond the terpene exposure that triggered its production. The cell manufactured the weapons during the walk. The weapons remain in the armory long after the manufacturing stimulus cleared. Well, the 7-day number reframed my own weekend hike. The walk I had been treating as recreation was producing an immune enhancement measurable a week later through proteins I could neither see nor feel being manufactured inside cells I could not detect. The calm I attributed to the scenery was real, but secondary. The primary effect was molecular terpenes crossing membranes, entering cells, upregulating genes, arming granules, enhancing surveillance. The hike was a medical intervention I had been classifying as leisure. Did the research isolate the forest air as the variable? 3 studies systematically isolated the active variable. Ching Li at Nippon Medical School designed the progression. Now, the evidence. Study 1, 2006. 12 healthy males walked 2 to 3 hours per day for 3 days under forest canopy. NK cell activity increased 56%. NK cell number increased 50%. All 3 killing proteins increased measurably. After returning to urban life, NK cell activity remained significantly elevated. 30 days post-return, still above baseline. Lee and colleagues documented these changes in the International Journal of Immunopathology and Pharmacology. Study 2, 2008. Identical walking protocol through an urban environment, same duration, same intensity, same subjects, zero significant change in NK cell activity or number. The walking was not the variable. The intensity was not the variable. The relaxation was not the variable. The break from screens was not the variable. Every psychological explanation for the forest benefit was present in the urban walk, the exercise, the outdoor air, the change of scenery, the social component. None produced the immune enhancement. The forest air was the only variable that differed, and the forest air was the only variable that correlated with the NK cell response. Study 3, 2009. Subjects slept in hotel rooms with vaporized hinoki cypress stem oil diffused into the air. No forest, no walking, no visual canopy, no bird song, just terpene inhalation during sleep. The subjects breathed air containing the terpenes forest's release, delivered through a commercial diffuser in a controlled indoor environment. Significant increase in NK cell activity. The terpenes alone were sufficient. The hotel room study is the definitive isolation experiment. It eliminates every confound except the chemistry. Just molecules in the air entering the lungs during sleep. The NK cells responded to the molecules regardless of the delivery environment. The walking was irrelevant to the immune mechanism. The visual environment was irrelevant. The acoustic environment was irrelevant. The inhaled chemistry was the active variable. Isolated from every confound, the first 2 studies left open. 3 studies. 1 variable isolated. The progression is the model of scientific rigor, observation in the field, controlled replication eliminating confounds, laboratory isolation confirming the active ingredient. Lee built the evidence the way an engineer builds a proof, hypothesis, test, control, isolation. The conclusion survived every confound elimination the research design imposed. Chemistry is sufficient. The forest provides additional channels the chemistry does not require, but the body receives. The lungs absorb the terpenes, the eyes absorb the something else entirely. What does the visual environment under the canopy do to cortisol? And then, the visual channel. Fractal dimension. Natural visual scenes, branching trees, leaf edges, dappled light filtering through canopy layers, bark textures, fern fronds, have a fractal dimension of approximately 1.3 to 1.5, self-similar patterns repeating at multiple scales. A tree trunk divides into primary branches. Each primary branch divides into secondary branches. Each secondary branch divides into tertiary branches. Each tertiary branch divides into twigs. Each twig produces leaves with veins that branch in the same pattern at a still smaller scale. The branching geometry repeats across 5 or more scales of magnification. The mathematical property that describes this repetition is the fractal dimension, and for natural branching patterns, the dimension consistently measures between 1.3 and 1.5. The mathematical property is measurable and specific, viewing fractal patterns at approximately 1.3. The dimension of natural scenes produced measurable cortisol reduction and alpha wave increase, as Taylor and colleagues at the University of Oregon measured, compared to geometric patterns at fractal dimension 1.0. The dimension of urban architecture with its right angles and flat surfaces. The evolutionary hypothesis, human visual processing evolved under fractal natural environments for millions of years. The visual cortex is calibrated to process patterns at fractal dimension 1.3 because that is the dimension of the environment the visual system developed in. Urban environments at fractal dimension 1.0 present a visual pattern the cortex was never optimized for, producing sustained low-level stress through the processing effort the unfamiliar geometry demands. The cortisol reduction from viewing fractal patterns at 1.3 may represent the visual cortex returning to its baseline processing mode, the mode it evolved to operate in, rather than the effortful processing the urban geometry demands. The forest removes a stressing input the urban visual field continuously imposes. Taylor published this in Nonlinear Dynamics, Psychology, and Life Sciences. The cortisol pathway the belly fat discussion described, from 11-beta HSD1 through the portal vein to hippocampal dendrite retraction, is reduced through a pathway the forest provides independently of the terpene chemistry. The fractal dimension of the visual environment under the canopy reduces cortisol through the visual cortex. The forest lowers cortisol through the eyes while enhancing immunity through the lungs. The two mechanisms operate through completely independent pathways. The terpene pathway runs through alveolar diffusion, bloodstream transport, NK cell entry, and gene expression. The fractal pathway runs through retinal pattern detection, visual cortex processing, and hypothalamic pituitary adrenal axis modulation. Neither pathway requires the other. Neither pathway interferes with the other. Both operate simultaneously from one environment, and you receive both without knowing either is active. Two independent interventions from one environment through two sensory channels operating simultaneously. A third channel is operating at the same time, and this one works through the brainstem. How does the forest soundscape reset the nervous system? Natural soundscapes. Birdsong at 2 to 5 kHz. Wind through leaves producing broadband pink noise. Flowing water with irregular temporal patterns. These acoustic inputs bypass the acoustic startle reflex. The brainstem processes them without triggering the amygdala-mediated sympathetic activation, that sudden high-amplitude urban transients, horns, sirens, construction impacts, produce within milliseconds through the inferior colliculus. The startle reflex is pre-attentive. The sound reaches the amygdala through the brainstem pathway before the auditory cortex has time to identify and evaluate it. You cannot choose not to startle. The sympathetic activation fires before the conscious brain recognizes the sound. A car horn, a siren, a door slamming, each triggers the same circuit regardless of whether the sound represents an actual threat. Urban acoustic environments produce dozens of startle circuit activations per hour. Each one adds its sympathetic pulse to the cumulative autonomic load the nervous system carries through the day. The forest eliminates all of them simultaneously. The exposure without a single startle circuit activation allows the sympathetic nervous system to stand down and the parasympathetic system to restore the autonomic balance the urban acoustic environment continuously disrupts. Restoration is measurable through heart rate variability, the beat-to-beat variation that increases under parasympathetic dominance and decreases under sympathetic dominance shows measurable increase during and after forest exposure. The contrast with the urban acoustic environment is stark. A city street produces sustained low heart rate variability through continuous startle circuit activation. A forest produces high variability within minutes of entering the canopy, as the brainstem registers the absence of threat signals and releases the sympathetic tone it had been maintaining in response to the urban soundscape. Natural soundscapes produce measurable parasympathetic increase and sympathetic decrease. Buxton and colleagues confirmed this in the Proceedings of the National Academy of Sciences in 2021, establishing that natural acoustic environments shift autonomic balance toward the parasympathetic state through the elimination of startle circuit inputs. Forest eliminates the acoustic threat signals the brainstem responds to involuntarily. The parasympathetic restoration is the removal of the sustained sympathetic activation the urban acoustic environment imposes through startle circuits you cannot consciously suppress. My understanding of the weekend walk shifted on the afternoon the three channels converged into one environment. The lungs were absorbing terpenes that upregulated NK cell gene expression for days to weeks. The eyes were processing fractal patterns that reduced cortisol through the visual cortex. The ears were receiving an acoustic environment that restored parasympathetic tone to the brainstem. Three sensory channels delivering three independent physiological interventions simultaneously. The dose response is specific. 2 to 3 hours produces measurable NK cell increase from a single visit. Overnight camping amplifies the effect through extended terpene exposure during sleep. 7-day persistence suggests one weekly forest visit maintains elevated NK cell function continuously. If you walk in a forest every Saturday, your immune surveillance is enhanced to every Friday. A weekly vaccination that requires no needle, no appointment, and no side effects beyond the calm you already wanted from the walk. Urban parks with mature trees deliver lower but measurable terpene concentrations. A daily lunch walk under old oaks provides a fraction of the forest dose accumulated across the workweek. The practical protocol from the evidence: one forest visit of 2 to 3 hours per week, ideally in a dense coniferous or mixed forest under closed canopy during the warmer months when terpene concentrations peak. The persistence means the enhancement from Saturday's walk overlaps with the next Saturday's walk, creating continuous elevated NK cell function from a weekly 2-hour investment. The evidence does not support the same enhancement from viewing photographs of nature, listening to recorded birdsong, or diffusing concentrated plant oils, although each may provide a partial benefit through the individual channel it activates. The full 3-channel convergence requires the physical environment, the air carrying the terpenes, the visual field carrying the fractal geometry, and the acoustic environment carrying the parasympathetic restoration. 3 sensory channels delivering 3 independent interventions simultaneously from the walk under those trees. The walk felt like one experience. The body received 3, the terpene chemistry enhanced immune surveillance through gene expression that persists for days to weeks. The fractal geometry reduced cortisol through visual cortex processing that operates for the duration of the exposure. The acoustic environment restored parasympathetic tone through brainstem circuits that respond within minutes of the startle input elimination. 3 timescales, the week-long window for the immune effect, hours for the cortisol effect, minutes for the autonomic effect, all 3 initiated by the walk. All 3 operating through independent sensory channels, the body processes without conscious awareness. The exposure under a canopy, 3 sensory channels, 3 independent mechanisms. The air carried terpenes that crossed the alveolar membrane, entered the NK cells, and upregulated the genes encoding the killing proteins those cells use to destroy infected and malignant targets. The visual field carried fractal patterns that reduced cortisol through the visual cortex. The acoustic environment eliminated the startle inputs the brainstem responds to with sympathetic activation. 7 days later, the killing efficiency was still elevated from a single exposure. The forest was delivering 3 measurable interventions through 3 channels. The body used without knowing any of them were open. The first tree discussion taught what happens at the surface, electron cloud repulsion across a gap the skin and the bark never close. What enters through the lungs is what this walk delivered. Both happened simultaneously during one encounter. The trees were always doing both. 4 channels from one encounter. The air delivered the chemistry. The visual field delivered the fractal geometry. The acoustic environment delivered the parasympathetic restoration. The bark delivered the electromagnetic contact. The forest was a 4-channel intervention the body received through 4 sensory systems simultaneously, each channel operating through an independent mechanism, each mechanism producing a measurable outcome the urban environment cannot replicate. Whether forest exposure should be incorporated into your health practice alongside the other interventions your physician has recommended is a conversation informed by the evidence Lee's studies established. The physics teaches what the air, the visual field, and the acoustic environment each deliver. The evidence quantifies how long the effects persist. The weekly 2-hour investment is the most efficient immune enhancement protocol the evidence supports from a single behavioral intervention.